CRISPR, a technology that has the potential to fundamentally change medicine. This gene scissors allows scientists to precisely alter DNA, tackling genetic diseases at their roots. What once sounded like science fiction is now a reality. But how does CRISPR actually work? CRISPR is a tool scientists use to precisely edit DNA. It works like a gene scissor that cuts DNA at specific locations. The technique has two main components: a guide molecule that locates the target DNA and the Cas9 protein that makes the cut. The guide, or guide RNA, is a piece of RNA that matches a specific DNA sequence. When the guide finds its target, the Cas9 protein binds to the DNA and cuts it exactly at that point. After Cas9 makes a cut, there are various ways to repair the DNA. Sometimes, new DNA segments are added to correct genetic defects. In other cases, faulty genes are simply removed. This method allows not only for the alteration of defective DNA, but also for the addition of new functional genes. This precision and flexibility make CRISPR revolutionary, targeting specific genes with fewer side effects compared to older gene therapies. What makes CRISPR special is its accuracy and ability to edit multiple genes simultaneously. Traditional gene therapies are often less precise and can only target one gene at a time. However, CRISPR uses the natural mechanism of bacteria that defend against viruses and has proven to be highly versatile and precise. Scientists are also developing new proteins like Cas12 and Cas13, which are even more flexible and can target RNA in addition to DNA. This makes CRISPR one of the most promising methods in gene research. In 2024, the first CRISPR therapy for sickle cell anemia was approved. This disease causes red blood cells to take on an abnormal sickle shape, leading to pain and complications. CRISPR therapy targets and modifies the faulty gene, allowing red blood cells to regain their normal shape. Patients can live with less pain and no longer need regular hospital treatment. However, this is only the beginning, as similar therapies for diseases like beta thalassemia are also being researched and show promising results. CRISPR can do more than treat genetic diseases. Currently, research is underway on a therapy that could treat type 1 diabetes, an autoimmune disease in which the immune system destroys insulin-producing cells. Through targeted CRISPR use, these cells could potentially be restored, eliminating the need for daily insulin injections. CRISPR is also being tested for amyloidosis to reduce harmful protein deposits that strain the heart and nervous system. These potential applications show that CRISPR is versatile beyond genetic diseases and could potentially help millions. CRISPR goes beyond hereditary diseases and could revolutionize the treatment of antibiotic-resistant infections. Some bacteria, like E. coli strains causing urinary tract infections, are resistant to antibiotics. Researchers are currently testing CRISPR-modified bacteriophages, viruses that specifically target these infections. If successful, this method could offer an alternative to antibiotics and fight antibiotic-resistant infections. With all these new possibilities comes great responsibility. One day, CRISPR could modify genes in the so-called germline, genes that affect not only the individual, but also future generations. Such changes could become permanently embedded in human genetics, laying the foundation for an entirely new kind of inheritance. The term designer babies describes the idea that parents could customize their children's genetic traits, for example, physical characteristics, intelligence, or other abilities. However, many ethicists warn that such interventions could deeply impact society. There is a risk that only certain people would have access to such technologies, creating a genetic divide. The idea of a society where only the wealthy can afford genetic advantages raises questions of justice and equality. Many scientists and ethicists are calling for international regulations and ethical guidelines before CRISPR is used on a large scale. The technology must be used responsibly to prevent unforeseen consequences and to ensure that access remains fair and equal. Only in this way can CRISPR truly be used for the good of all. CRISPR has already shown how powerful gene editing can be. The technology has the potential to cure diseases and fundamentally change medicine. But alongside progress, it carries responsibility. The future of medicine is on the brink of a revolution. What opportunities and risks do you see in CRISPR's future? Share your opinion in the comments. If you enjoyed the video, give us a thumbs up and subscribe to our channel. Thanks for watching, and see you next time.